Okay, we will discuss Chapter 39, Automation in Hematology. Now we know automation is firmly established within every hematology lab. And from the previous class, we have had many printouts, so we know that they can perform a CBC, which includes a 5-part differential and an absolute reticulocyte site count, and some can even perform body fluid. Now there are a number of principles for cell counting, and we're going to discuss them in the next slides. I'm going to discuss two of the major principles in hematology instruments, which are impedance and optical light scattering, or laser optical. So first, the impedance principle of blood counting is based on the increased resistance that occurs when a blood cell with poor conductivity passes through an electrical field. So here, the number of pulses indicates the blood count, and the amplitude or height of each pulse is proportional to the volume of the cell. Some examples of companies that use this principle are Beckman Coulter and Sysmex and Abbott Diagnostics. Now the second principle of optical light scattering, or laser optical, is based on the light scattering measurements obtained of a single cell that passes through a beam of light or laser. Now, this cell creates a forward and side scatter that photodetectors detect, and the degree of forward scatter is a measure of the cell size, and the degree of the side scatter is a measure of cell complexity or granularity. So Siemens Healthcare also uses this type of principle for certain types of blood counting, and when we get to the CBC report, I will show you which types of analytes within the CBC report determine the different types of principles. So here's an example of a Beckman Coulter LH750 printout where you can see that the Coulter uses the principle of impedance as well as a variation of optical light scattering for leukocyte differentials and reticulocyte enumeration. So seeing here in the Wbc histogram, we're using the principle of impedance, and as the cells pass through, the number of pulses indicates the cell count, and the amplitude or height is proportionate to the cell. So this first peak here are lymphocytes, the second peak are monocytes, and this last group are bunched into granulocytes altogether. Now the Wbc scatter plot below uses optical light scattering principles, so each of these dots represents a cell within the group, and as bloods pass through a laser or optical beam of light, they create a forward scatter and side scatter that determines the complexity and measurement of the cell. So you can see this first group here are lymphocytes, and now behind this you can't see basophils, but in the 3D image they're colored and you will be able to visually see them. The one above are monocytes. This larger darker group are neutrophils, and you can see the darker the group, the more cells are within the group. And then this lighter category are eosinophils. And in the bottom here to the left is a nucleated red blood cells, or some kind of ghost population or debris that has to be looked at under the microscope. Begin by dissecting the Coulter LH750 report. You can see it is classified into multiple categories based on the type of cell we're looking at. So in the beginning we have a total white blood cell count and its 5-part differential underneath. This percentage is equivalent to the relative value, or how you would count up to 100 in your differential. Okay. Now it will also do a nucleated red blood cell percentage. As these nucleated cells, the machine thinks it is a lymphocyte. But on the scattergram, you can see that it is very small in size and complexity, so you can easily see that there is some kind of abnormality within the scatter plot, and you will see nucleated red blood cells in your differential when you're looking at your slide. Now, there are 5-part absolute count as well from your relative counts that you need to pay attention to based on the patient condition. So we will be calculating these values ourselves. So from the relative values, you will get your absolute value. So the equation for that, for example, your neutrophil relative value would be turned into a decimal, so 0.549 multiplied by the total white blood cell count of 8.3 will give you a neutrophil absolute value of 4.6. Now these values will tell us what's going on with the patient condition, and that will help the physician when we're talking about treatment. The second portion here, you're used to seeing a total red blood cell count, as well as the hemoglobin and hematocrit portions to tell us if we have some kind of anemia happening. From these, we can classify the type of anemia from our red blood cell indices by calculating the MCV, MCH, and MCHC values. The RDW value tells us the number of sizes or different variations in sizes of our red blood cell, and that can also be seen on the red blood cell histogram. The third component of a CBC would be the platelet count, and you would also see an MPV, which is similar to the RDW for the red cell parameters. For the platelet parameters, it will also show in the histogram and will give you a sense if there's variation in size in platelet morphology. The last portion of the culture would be the reticulocyte count, where it will do a relative and absolute count as well as immature reticulocyte fraction. Okay, now let's talk about what happens inside the instrument when a sample goes through. So for these, it begins with a closed EDTA sample that goes through that's divided into 4 different aliquots. So the first aliquot is delivered to the red blood cell platelet dilution chamber. Here these red cells are diluted with an electrical conductive diluent. Within the chamber there are 3 separate apertures, each with an internal electrode. So as the cells pass through the aperture, there's increased resistance, so that would be the pulse height, between the external and internal electrodes that's proportional to the cell volume. Now there's always a steady stream of diluent, so these cells they're prevented from re-entering the aperture, so it flows out so they're not counted again. Okay, so any particle or cell of more than 36 femtoliters are counted as red blood cells. Now because these apertures are located within the chamber, there are 3 different counts obtained. So if these counts compare, then the system will report out a red blood cell count. Now this will produce a red cell histogram, which is a size distribution curve. Okay, so it's created based on the cell volume and cell number. The red cell histogram allows you to visually see the changes in size within the red blood cell. So for example, if we have a normal size of cells and there's a shift to either the left or right, you can see within the histogram if there's any red cell disorders, if the cells are going to be smaller or if the cells are going to be larger. So it's like a double check. The instrument can directly measure the red blood cell count, the MCV, and the RDW. In the calculated counts based off of these, it will calculate the hematocrit and the MCV. Now the platelet count is also obtained from this chamber, and any particle or cell between 2 and 20 femtoliters are counted as platelets, and a platelet histogram is obtained, as well as a mean platelet volume. Okay, a second aliquot is taken and delivered to the white blood cell hemoglobin chamber, where the leukocyte count and hemoglobin determinations are made. So here the white cell count is directly measured by impedance from the dilution after lysing all of the red cells. So as the red cells lyse, they release hemoglobin from the cells. Now here hemoglobin is converted to cyan methemoglobin and shrink the leukocyte cell membrane in cytoplasm. Therefore, the Wbc count represents a measure of the cell volume rather than the native size as it passes through the aperture. That's why it looks different on the histogram. So particles larger than 35 are counted as leukocytes. Similar to the red blood cell, white blood cells are obtained from 3 apertures as well, and the average, if they are consistent, the instrument will release this average count. So a WBC histogram represents the distribution curve of the data that allows for visualization of subpopulations based on their relative sizes. So the WBC histogram is the basis of a 3-part differential identifying lymphocytes as cells between 35 and 90 femtoliters, mononuclear cells as cells between 90 and 160 femtoliters, and granulocytes as cells between 160 and 450 femtoliters. The third aliquot goes into the orbital mixing chamber, which determines our 5-part leukocyte differential using a volume conductivity scatter flow cell. This flow cell, so it only uses one cell at a time and detects one cell at a time, which is called hydrodynamic focusing. So as each cell passes through the flow cell, there are 3 separate measurements taken that determine volume, conductivity, and light scatter characteristics. And the scatter plot is determined here. Each of these dots are a cell. So we have lymphocytes, and in the Coulter, basophils are behind here so you cannot see them. But we also have monocytes, neutrophils, and eosinophils. Now here, if nucleated red blood cells are suspected, they will be determined by the computer system by looking at the white cell histogram that's generated through impedance and presence of cells to the left of the lymphocyte. If both plots indicate that nucleated red cells are present, the computer system will evaluate the combination to rule out giant platelets or small lymphocytes. And if all the data point to the measured presence of nucleated red blood cells, the number of nucleated red blood cells per 100 is derived from the data calculated or collected by VCS technology and then plotted here. The fourth and last aliquot is sent to a heated reticulocyte dilution chamber where it is mixed with new methylene blue, and you can see the residual RNA precipitated within the reticulocytes. So it will give you a percentage, an absolute, and then it will send it to the VCS flow cell for analysis, and if there's light scattered data generated, you will also get a scatter plot. Now this sophisticated technology of VCS can also determine the difference between immature reticulocytes and maturing reticulocytes. So it will also give you an immature reticulocyte fraction. Here's a clearer demarcation of what a Wbc scatter plot looks like in a Beckman-Coulter analyzer. So here it is plotted volume against light scatter, where you can see that there are separate leukocyte populations here in a two-dimensional space. So on the computer screen, all of these different populations will be in color, and you will also be able to visualize the basophil population that's located behind lymphocytes. So here you can clearly see what a red cell histogram and count look like. So here only the shaded areas of this curve are used to detect the RDW. The other areas not shaded represent things like large platelet or clumps or any kind of interference within the machine that is not used in the actual count. Okay, now let's dissect another one of the major manufacturers of hematology instrumentation, Sysmex. Okay, so Sysmex incorporates 4 different technologies, including impedance, radiofrequency, absorptive spectrophotometry, and flow cytometry with dyes to determine the CBC, 5-part diff, NRBCs, and reticulocyte count. So an aliquot of this specimen is taken to be used for the red cell and platelet counts that are determined using impedance similar to the Coulter instruments. So the red blood cell count represents the number of cells that fall between 25 and 75 femtoliters as a lower discriminator, and a higher or an upper discriminator of 200 to 250 femtoliters. So likewise, the platelet count is determined between cells at a lower discriminator of 2 to 6 and an upper discriminator of 12 to 30 femtoliters. In Sysmex analyzers, hemoglobin is measured in its own channel using the sodium lauryl sulfate method. So here a portion is diluted with a lytic agent where it converts ferrous iron to ferric iron, forming methemoglobin, and this is measured through spectrophotometry. So there are 2 separate channels, the diff and the Wbc baso, that are used to generate the total white blood cell count and the 5-part differential. So in the diff channel, a portion of the blood sample is aspirated and then a lytic agent is added to remove the red cells, and it also creates holes in the membranes of the nucleated cells, white cells, allowing a dye to enter them, bind to the DNA and RNA, and then here, passing through a single laser beam, each cell generates forward and side scatter, and the intensity of that fluorescence depends on the cell type. So the diff scattergram represents the plot of side scatter versus fluorescent intensity. These instruments are special because it has an Immature granulocyte software that detects the immature gland population. It can count them as metamyelocytes, myelocytes, and promyelocytes and produce a scattergram plot. So for the Wbc baso channel, the specimen or aliquot is diluted and a special lytic reagent breaks all of the red cells, and it shears the leukocytes except the basophils and is added to the white cell baso channel. So the optical detector analyzes each cell's forward and side scatter as it passes through the laser beam. This adaptive cluster analysis evaluates the information to determine the total WBC count and basophil count. Now there's another channel, the Immature Myeloid Information Channel. Here They act selectively on lipid membranes. Here, mature leukocytes with phospholipid-rich membranes are lysed, leaving only the bare nuclei. So the immature myeloid cells remain intact, and then these are measured by direct current and radio frequency to determine the degree of immaturity and plotted on its own scattergram. Now, in the nucleated red blood cell channel, cells pass through here and are diluted. The red cells are lysed, and there's a fluorescent agent that is added, which binds to the nucleus of the cells. So the reagent only perforates the membranes of leukocytes, allowing the dye to enter the cell and bind to the intracytoplasmic organelles, but it does not alter the leukocyte's shape. So the leukocytes exhibit a stronger fluorescence than the nucleated red blood cells. So as the nuclei of these NRBCs are intact, the leukocytes pass through the laser beam of the optical detection block, and they generate forward and side scatter and fluorescence. So this information is used to create the NRBC scattergram, which plots fluorescent intensity versus forward scatter. The reticulocyte count is determined by the RET channel. So within here, the fluorescent dye penetrates the cell membranes and stains the blood cells and binds to residual RNA within reticulocytes. So the data uses the forward and fluorescence information to generate this scattergram, and from this scattergram, cells are classified as mature red blood cells, reticulocytes, and platelets. The reticulocytes can be further classified based on their intensity to determine their maturity. Now here the platelet count from the RET channel is designated as a platelet optical count, and is compared to the platelet count from the RBC platelet distribution. So if the platelet count is extremely low, the count from the RET channel is reported because the result is more accurate. And you are used to seeing the scattergram from the Sysmex series, and they're labeled as their colored classifications based on their scatter plot. So this semester, in place of identifying lymphocytes, monocytes, neutrophils, basals, and eosinophils, we are also going to be seeing and discussing the immature granulocyte fraction of the scattergrams, as well as be able to identify debris. So here you can visually see on the NRBC scattergram how in the NRBC channel that nucleated red cells are clearly delineated from the red cells based on their lower frequency or intensity of fluorescence plus their smaller size.